So, good morning or good afternoon, everybody. It's my pleasure to continue our Global IMMUNOTALK series by introducing another spectacular speaker, Nicolas Gaudenzio. Nico is a research director at the French National Institute of Health, INSERM, and is the chief scientific officer at a biotech company called Genoskin that's located in Toulouse and in the United States. Nico received a master's in bioengineering and a PhD in immunology at the University of Toulouse in France. He completed his postdoctoral training at the University of Stanford in the US. His work has contributed substantially to identifying molecular and cellular targets involved in allergic and non-allergic inflammation. He leads currently a large research program dedicated to investigating the role of neuroimmune interactions in health and disease. He is, and that's very spectacular, a three-time laureate of the European Research Council. So he received a starting and consolidator and a advanced ERC grant, which is spectacular. And he has received international prizes such as the Acteria Early Career Research Prize, which is given by the European Federation of Immunological Societies. He has a Rising Star Award by the International Union of Immunological Societies, and he's a member of the European Academy of Allergy and Clinical Immunology. And I think there's many other things I could mention, prizes, awards, research grants, and so on, but that's not the point of our meeting. We want to hear your spectacular work, and it's really a pleasure to welcome you and to have you and to join us, and we look forward to your talk. But at the beginning, we usually have a short question that we have agreed upon before. And so the question we have chosen for today is, I think, a very important one, and a very important one for society and for scientists, or that deals with a society problem. And that is, how can you as a scientist balance family life and work life and your career, and what advice would you give to people who are on the track and try to achieve both things successfully?
Thank you, Dietmar, for the very kind introduction. And yeah, I think this question is super important, and because I usually tell to myself, at the end of your life, what do you think if you think back of what you have been doing during your life? So it's very important to have discovered how one receptor on mast cell or on neurons works perfectly, but at the end, I think it's super important as well to be happy of your personal life. And I can share my personal perspective on that. So I have, for example, two kids and one wife, and at a lab of around 20 people. So, and when you have a lot of postdocs, they produce many data that then you need to keep up with that, and it can take a lot of time. So I usually tell to myself from 6:30 p.m., to at least 9:30 p.m., it's a dedicated time to the family. So I shut down my phone, my email. Today is an exception for me because we're going to be passing the deadline.
It's only
But
6:30 yet.
maybe I didn't tell you, but I'm going to shut down everything in 30 minutes. But yeah, and I have this time of dedicated truly to the family where I do not respond to anyone for during the week, and obviously trying to manage that during the weekend. So that's my personal feeling on it, and I don't know how other people are doing.
Well, it's something that sounds a little familiar. I'm not turning off the phone, but otherwise it's the sort of similar period. No, perfect. No, very, very good. And it's very good that we discuss also these topics and not just science. But speaking of that, now I would like to see your spectacular work. And thanks again for joining us. And please share your screen now.
You're welcome. Okay. Do you see the full screen?
It looks very good.
Okay, thank you, Detmar, and I'm really happy to be here, and thank you so much for the invitation. So I'm going to try to keep my presentation within a 40 minutes time frame. So we're going to go from some data that are already published up to now to 2024, and some unpublished data also that I'm going to share with you. So this is my disclosure. And so my lab has a strong focus on mast cell biology. And, mast cells have been underestimated for a really prolonged period of time by immunologists. And what we know now is that they are involved in many, many processes. In addition, that only IgE-dependent allergic reactions. Here you have mast cell. It's a human skin section analyzed by 2-photon microscopy, and you have mast cell here located all around blood vessels here. They are also obviously around neurons, and those mast cells, when they are in the tissue, they basically are sitting there. They don't move. They do not migrate at steady state, at least. And this is, for example, just intravital imaging, where you have the blood flow in green and you have mast cells sitting around here in red. In blue are the collagen fibers. And when you trigger an IgE-dependent reaction, what you will see is that there will be a strong extravasation in the tissue due to mast cell mediators, and you will see the mast cell granules that will be then floating around within the tissue and even taken up by the bloodstream. In 2015, the group of Xing Dong and Johns Hopkins University has discovered that the receptor MRGPRB2 or X that's in the mouse or X2 in human are really important receptor that has completely changed the way that we perceive mast cell biology now. We know that they are involved in many physiological and pathological processes through interaction with neurons, thanks to this neuroreceptor. For example, B2 or X2 can bind many molecules. It can bind neuropeptides, as I just said, such as the substance P. This is the one that will be interesting for us today. But it can also bind antimicrobial peptides and bacterial products. And unfortunately, many synthetic drugs involved in pseudoallergic reaction and injection site reaction. And we are also working on that via Genoskin side. I'm not going to cover that today. So when you engage MRGPRX2 and B2, what you get is a massive release of granule-associated mediators, histamine, heparin, many proteases, but also lipid mediators, cytokines, and chemokines. In 2019, the work done by Nadine Serran, who was a PhD student in the lab, showed that actually sensory neuron and mast cell in the skin were really in close opposition. Here you have the neurons in blue, the mast cell are in red, and it's very hard to find a mast cell that is not in contact with neurons. And what we found is that those mast cell and neuron form what we name sensory cluster, capable of detecting allergens and certain protease activity of allergens. And when you engage this receptor, you get mast cell degranulation, and this is actually required for the development of type 2 immunity in the context of models of eczema. What we show is that we had a model of eczema in which we apply dermatophagoides farinae and a toxin from Staph aureus. So these are two antigens found in the surface of the skin of patients. And when you do that epicutaneously on the mouse, you will see that you develop strong inflammation, dysregulation of the epidermal barrier. You get house dust mite, specific type 2 immune response, and features of atopic dermatitis. But if you use a mouse that lacks sensory neurons, or a mouse that lacks substance P, or the mast cell deficient animal, and an animal in which you have a normal number of mast cells, but for which MRGPRB2, so the receptor for substance P on the surface of mast cell, is missing, all those 4 animals were completely protected from the development of the pathology. So this really prompted us to really realize that MRGPRB2 in the mouse and certainly X2 in human actually could be a nice target for the development of therapeutic drugs. And in this movie, what you see is that when we directly, and I can rerun it, when you directly activate sensory neuron in the skin, the mast cell nearby start to degranulate. So there is really a bidirectional communication between substance P producing neuron and MRGPRB2 positive mast cells in the skin. So we published that in 2019, and the very same year, the work performed, some of it in collaboration with my lab, by 2 very talented PI, which are Brian Kim and Xing Dong, also show that the very same interaction between mast cell and sensory neuron would be also really important for driving pain sensation, non-histaminergic itch as well in model of allergic inflammation, and also in fighting against bacteria. So this really, in 2019, paved the way toward the development of molecules that would block actually this receptor in human. In this context, the company Escient Pharmaceutical, which is now a part of Inside, actually contacted us as they were generating lead candidates actually to block MRGPRX2, and in particular this one named EP262. And we actually helped them to validate this antagonist and to have the proof of concept that it works actually in the mouse and humanized animal and the human context. And so the idea was that if we can block MRGPRX2, we could block sensory neuron mast cell interaction and maybe really prevent the development of many inflammatory disorders that are linked to the skin and associated with pain and pruritus as well. So this is part of a recently published work in collaboration with this science, in which there is the full validation of the antagonist. I encourage you to read the article, and it's fabulous work done by Wolam et al. And here, what you see are actually transgenic mouse that are for which MRGPRB2 has been removed and has been replaced by the human version MRGPRX2. So it's a humanized mouse for this receptor on mast cell. And what you can see in blue on the top is actually the Evans blue extravasation. So when you inject an MRGPRX2 agonist, you will trigger mast cell activation and will then trigger the release of blood vessel content. And this is what we can see here in blue. Here you have a PBS where you have obviously no extravasation, but when you inject an agonist of MRGPRX2, substance P, PACAP, which is also very important, a neuropeptide secreted in the brain as well, MBP, catibant, or a synthetic compound and compound 4880, you trigger indeed extravasation all the time. But if you pre-inject the EP262, you can see that we completely abolish the development of these mast cell dependent reactions. What you can see here are human primary mast cells that are incubated with fluorophore, which is a calcium tracer. And here you can see that when you add substance P, you trigger MXGPRX2 and you have a massive release of granules here in red. However, when you do the very same experiment in the context of pretreatment with EP262, what we could see is that we could completely block the activation of human primary mast cell, and they have been doing also additional experiments in human skin explants as well. So now this molecule has undergone a phase 1b in which it has been very well tolerated. It's an oral administration. And now there are two parallel phase 2 studies on chronic spontaneous urticaria and atopic dermatitis. And we are really looking forward to see those results. So coming back to more basic science a little bit. So when we published this data, we actually figured that we don't know that much about those MRGPRB2 positive mast cell and in mast cell in general in terms of ontogeny and transcriptomic heterogeneity. And so this is the work performed by Lilian Basso and Marie Taubert. It was published last year in collaboration with talented people from France and Scotland. And what we started to do is a pan-organ single-cell profiling of mouse mast cell. And it's very easy to isolate mast cells. So we had our own data set, and then we aggregated data set from other published data. And it's very easy to isolate mast cells as I express CPA3. You can also do that on the co-expression of c-KIT, the high-affinity Ig receptor as well. And when you do that, what you see is that you will have on one side mast cell that will be in most of the tissue that will aggregate on the right side, and then the mast cell from the gastrointestinal tract that will then segregate apart on the other side. This is what is named for many years connective tissue type mast cell and mucosal mast cell. But what was very interesting for us is that if we look at the expression of MRGPRB2, we found that it's actually not only restricted to the skin tissue or the peritoneal cavity, but it was a global marker of mast cells that are found in most of the tissue but the gut. However, if you take, for example, the protease MCPT1, we found that it's an exclusive marker of those mast cells that you can find in the gut. We then perform additional experiments just to validate those data. We found that indeed in the skin, you find only B2 positive mast cell. However, if you look at in the guts and gastrointestinal tract, you will find mainly here in green, actually a B2 negative mast cell, but you can still find that some population that we did not capture by single cell RNA-seq found in the muscular layer of the gut that can express actually B2, and we still don't know their function. So when we analyze the transcriptomic profile of those mast cells, we could define a very clear transcriptomic signature for every type that was conserved across tissue for the two types of mast cell. The CTMC, so the connective tissue type mast cell, would express high level of MRGPRB2, also MRGPRB1, for which the function still to be fully explored, but also additional markers such as ApoE, CPA3 high, the kinase 1 and CD81. However, those mast cells that are found in the gut, they are B2 negative. They express super high level of MCPT1 and MCPT2 protease, many integrins that we think that this is for them to be anchored to the epithelial layer. They are really at the interface between the epithelium and the microbiota. So we still don't know their function, but they might be a strong link there. And also what was very interesting for us is that they express quite good level actually of CX3CR1, which was before associated really with macrophages. And if you take a CX3CR1 GFP mouse, here the GFP is in red, you co-stain with MCPT1, which is an exclusive marker for mast cell, you will find that many of the cells actually co-stain in the gut mucosa between MCPT1 and CX3CR1 GFP. So we need to be really careful if we use CX3CR1 DTR mouse in the gut, actually, because we will be depleting most of the mast cell, actually, and this could have difference in the interpretation of the results if we are studying macrophages. So I'm not showing you all the detail here, but what we found is that, and that was also previously confirmed by the group of Marc Bajenov as well in the CEML in France, is that mast cells that are in the skin, and what we show is that all the B2 positive mast cells, they come from the yolk sac, they derive within the embryo. They will then seed all the tissue within the embryo and stay there actually for a really prolonged period of time. We found that they are at less there for at least 6 months before being replaced, and they are not replaced at least a steady state by bone marrow progenitor. So we still don't know what are the progenitor to replace them. And they have a specific core signature, and you will find them in most of the organs. And what was really interesting as well is that in any organ that you can look at, they will also be found in close contact with the neurons, such as in the ears, in the ear here, the tongue, the trachea, the lung, the hair, and also in the muscularis of the GI tract. On the other side, you have those B2 negative mast cells. Those B2 negative mast cells, they are not developing within the embryo. We found that they are there only at birth. They are progressively developing. They are dependent on both microbiota-derived signal, which is not the case for the B2 positive mast cell for development. But here, if you use a germ-free mouse or if you treat with antibiotic, you actually reduce of more than 60% the population of mucosal mast cell in the gut. So they do not express B2. There is a constant turnover of those mast cells by bone marrow progenitor after at least 1 month, and they are coming from a progenitor, and we use fate mapper mouse that have been MS4S3 fate mapper mouse that has been developed by Florent Ginoux to see whether they were actually coming from GMP, because it's what is actually written in the textbook, but we didn't find any staining, any fate mapping in those populations, neither the connective tissue type actually. So we think that they are coming from an intermediate state between CMP and GMP that still need to be identified. So there is room there for research if people are interested. And they also have their specific core signature, and they will be found mainly in the stomach, in the gut, and also in the lungs. However, when you go in during a type 2 skin inflammation, what has been shown by the group of Kenji Kawashima last year is that when you do that, you have actually bone marrow progenitor that can be recruited to the site of inflammation, and they will not give rise to B2 negative mast cell, but truly to B2 positive mast cell. So during inflammation, you have a mixture between fetal mast cell of fetal origin and bone marrow derived mast cell that will then acquire the same phenotype with the mutation. So then when we switch to human, everything gets a little bit more blurry and much more complicated. So we aggregated many data sets also found in the literature and from the Tabula Sapiens, and we can also from almost 30 organs, and we could find a very clear, again, signature here of c-KIT, CPA3, and using tryptase and kinase as well to identify mast cell. And if you study mast cell from different data sets, I would encourage you to actually reanalyze data, because in some of the data set, mast cell unfortunately have been missed or have been aggregated with basophils, which are completely different cells. And when we do that, and again, you plot all those different mast cells, what we found is that the dichotomy between the connective tissue type and mucosal mast cell actually was completely unfound. We couldn't find this connective tissue versus mucosal mast cell, but we could find unbiasedly 6 independent clusters. So we don't know whether they are different state of mast cell population, different population, but they do coexist within different tissues. They also have specific transcriptomic program that you can identify. And so here, this is opening a new area actually to investigate how different subtype of mast cell in human at least can, influence the development state of pathology or regulate homeostasis in different tissues. And we could reconstitute the distribution of each population across the different tissues. The bladder is really highly enriched in mast cell. They express really high amount of MRGPRX2, for example, as much as in the skin. And what was also very interesting for us is that in the skin, for example, I could have taken another organs, but they express, you have at least the coexistence of 3 independent clusters, MC1, MC3, MC4. We think that they are, maybe one can be the activation statue of the other one. We're still digging into that. But it's very important to understand how different mast cell population will express different surface marker could be regulated during disease state, because it could really implicate drug development and find more personalized target for disease treatment or for targeting a specific organ. And a very small example that we recently published in 2024 in allergy, this is the work by former PhD student that is now an employee of Genoskin, Manon Scolar, is that, well, we started from the observation that during COVID vaccination, there are many pseudoallergic reactions that really look like MRGPRX2 engagement on mast cell. And this is an example with the Moderna vaccine that I'm going to show you here. So we took the biostabilized natural human skin from Genoskin that we injected with the Moderna vaccine. And I'm just going to give you a glimpse into it, and because it's already published and you can look at it if you want to. But when you do a single cell RNA-seq on which cell will actually incorporate the LNP particle, and it's easy, you just need to detect the spike sequence among the different immune cells within the skin tissue. And that was, if you do a sub-Q or an intradermal injection, it gives exactly the same results. We found that more than 60% of all LNP particles that can be detected after the injection are going into mast cell. So we still don't know how. We know that they are also slightly degranulated when you inject the vaccine. So I think these are many, many future therapeutic applications as well that we would like to investigate. So now for the remaining 20 minutes of my talk, I would like to switch to unpublished data. And those unpublished data, this is also the work by Nadine Serran, who worked previously on neuron mast cell interaction at the beginning of the talk. And it's about the relationship between prenatal stress and what we name pediatric eczema. So according to the EIACI, and we have the same data actually for the other part of the world, including the US, 50% of Europeans will develop allergic disease by 2050. And what was really striking is that among 100 babies that are born, in the same hospital, 20% of them, 20 of them will actually develop signs of eczema during the first 4 years of life, and even during the first weeks, days of life. And that was something that really was puzzling us. And we thought that there would be something additional than the microbiome, or that the sensitization with allergen actually right after birth that could happen. And if we take this graph of the onset of eczema according to the age, we were really interested by this very early onset of eczema with remission. Most of the kids, 60%, 70% of them will actually going into full remission around the age of 5, 6 years old. So that was really interesting for us. And so there were some small Scandinavian epidemiological studies that found that maybe maternal exposure to stress during pregnancy could increase the risk of eczema in kids. And so we wonder whether environmental perturbation in utero could predispose to pediatric eczema at birth. So for that, we switched to a mouse model, and this is in collaboration with Nicolas Senac at the IRSD in CERN in France as well, and in which we use a model of prenatal stress. So pregnant dams are actually exposed to bright light 3 times a day for 30 minutes and for 6 consecutive days between E13 and E18. After that, we go for analysis at birth, but also in young adults at 8 weeks old, which is more convenient actually to perform many experiments than in new pups. So when we do that and we analyze the weight of the pregnancy, the weight is totally normal, so they seem to have a healthy pregnancy. When you look at the litter size, it's totally okay. You cannot have, there is no difference male and female, and the pups are still gaining weight quite normally if they are coming from prenatal stress. So PS is if the pups are coming from prenatal stress mother or from control mother. When you look at the skin of those animals, again, at week 8 or at birth, the skin looks completely normal, normal epidermal thickness. So, and when you look at the percentage of the different immune cells, there is also a normal distribution of all the immune cells. However, if we look at a little bit the features of pediatric eczema, it often occurs at flexural location where the skin is exposed to constant mechanical frictions, such as here you can see on the foot, but also the cheek, the knees, or the elbows. So we thought to reproduce this mild, actually, mechanical friction. So for that, we developed a model of what we name light, wet mechanical friction, in which we apply on the surface of the skins of those animals. And this is why we use actually 8-week-old animals, because it's more convenient. A gauze that is embedded with either water or PBS, it works the same, and then occluded with a tegaderm. Then basically the gauze will slightly exert a small mechanical friction on the back skin of the animals. And we do that on animal from controlled pregnancy or from stressed pregnancy. And if you do that on animal from controlled pregnancy, obviously applying a PBS with a gauze pad doesn't do anything. There is absolutely no sign of inflammation. However, when we did that on pups that were coming from a prenatal stress mother, we found that they start to develop actually a very severe inflammation that resembles eczema. And here that we can measure very easily with an increase in the epidermal thickness. So we wanted to understand a little bit more about that. But what was interesting, and I told you at the beginning that around 6 years old, 5 years old, the kids that develop eczema, pediatric eczema, for most of them, they will go in full remission. What was also very interesting for us is that if you use 6 months old prenatal stress mice, they actually do not develop the pathology anymore. They're kind of, the phenotype also naturally reverses with the age. So we wanted to dig a little bit into that. So the fact that they actually are again more sensitive to mechanical friction prompted us actually to investigate whether they will feel more, mechanical pain, mechanical sensation on the surface of the skin. And for that, we used the von Frey filament assay on the hint pod. And what we found is that the von Frey filament threshold was dramatically decreased, meaning that they are more prone actually to find mechanical stimulation and mechanical pain at the site of stimulation. So when we know that mechanical pain or sensation is vehicled mostly by nociceptor within the skin tissue. So we stain with the beta tubulin that stain for most of neuronal fibers, but also with tyrosine hydroxylase, which in the skin, not in the foot pad, but in the skin, mostly stain from threshold mechanoreceptor. And this is how it looks in the control skin. But if you look at the skin from prenatal stress, pregnancy, and those pups, they have a hyperinnervation in terms of density of most of the neuron projections. So we then took the dorsal root ganglia of those mice, in which the cellular body of the sensory neurons is located. Then they project within the tissue. And we did a bulk RNA-seq analysis, and we found that there is more than 300 genes that are differentially expressed between prenatal stress offspring versus control offsprings, and with most of the genes that were dysregulated associated with CLTMR and non-peptidergic nociceptor, which are involved in mechanical sensory information conveying. And we also confirmed that those two populations were dysregulated at the functional level, because if you use some agonists that selectively activate those populations, you will find that the threshold activation will be much higher, while the peptidergic population was actually unchanged. So they seems to have a problem as well of mechanosensation and of at least non-peptidergic and CLTMR within the skin tissue. So we now studied the immune compartments. And so we perform a single cell RNA-seq analysis enriched on CD45 positive immune cells. We also have actually the keratinocyte compartment, and we didn't find any difference in the structure and the transcriptomic profile of keratinocytes in those mice. However, when we analyze the distribution of all the population, and this is the aggregated data set, but if you split the data set between prenatal stress or control mice, you will find exactly like the flow cytometry analysis at the beginning, that they have a normal distribution of immune cells. But what was very interesting is that if you look at the genes that are differentially expressed, what we found is that mast cells were profoundly impacted by this stress during pregnancy, with more than 500 genes that were differentially expressed, while it was not really the case with the other populations. When we look at the genes that are differentially expressed, and these are significant genes with an adjusted p-value, We found genes associated with granule exocytosis, activation, energy, metabolism. It looks like they have a kind of a hyperactivated mast cells with a weird granules morphology eventually. So when we look at how they look like those mast cells, if you take, and this is true at birth, and this is true in 8-week-old pups for prenatal stress, we found that as compared to control, when you look at in the skin of those pups, mast cells are already completely degranulated at steady state. And this is the quantification here. So we next wonder whether those mast cells that are impacted during pregnancy would actually be required for the development of this model of light friction. And indeed, we use a KIT deficient animal. So this KIT is a stem cell factor receptor, and those animals, they lack stem cell factor, they lack mast cell, they have other abnormality, but they are a good model of mast cell deficiency. And what we do see is that those mice were completely protected from the development of this phenotype of pediatric eczema after stress during pregnancy. We then confirmed those data also using MCP5 Cree mice, the IDTA. MCP5 is the protease expressed selectively by those B2 positive mast cells. And when we did that, we found that actually mast cell, the pups that lack MCP5 actually positive mast cells, so those skin mast cells were completely also protected from the development of the pathology. So you really need those skin mast cells to develop the disease. The skin mast cells that, if you remember during the first part of the talk, are actually coming, that are of fetal origin actually, and they are developing during the period that we are stressing the animals as well. So, so we also wonder, why would this phenotype actually reverse 6 months after? And we found this very nice article that was published in Nature in almost 50 years ago, in which they suggested, based on their experiment, that mast cell would live within the skin at least for 6 months before being replaced. And indeed, our data that we published in the GXMed last year also confirmed these results. We basically find, I mean, part of them, obviously, we find the same result, but just using more expensive experiment, which is a kind of frustrating. But 6 months after, we found that those mast cells started certainly to be replaced as well, not by bone marrow progenitor dose. And indeed, if you analyze, you do a bulk RNA-seq on those mast cells that you find in the skin at week 8 from prenatal stress versus control, you will find that they are really dysregulated. However, if you now take the mast cell that you will find after 6 months, you will find that those mast cells, they have a completely normal phenotype. They are undissociable from control pregnancy, meaning that those mast cells are not, subjected to this in utero inflammatory or in utero stressing environment anymore. And so those new mast cells that are coming after 6 months, they are basically not pathogenic anymore. And indeed, when you look at their face, you find that those mast cells at week 24, when you do not develop the disease anymore, are not degranulated anymore. So then we wanted to take a step before, and we look at what is going on in utero. So we then perform the same experiment of prenatal stress, and we harvested actually the pups, the embryos, I would say, at E18.5, just after the stress event before birth. And what we did is that we did a cross-section and an avidin staining of those embryos. And here you have the mast cell in red that you can find mostly actually at this age of time, mostly in the skin. This is macrophages and skin mast cells are developing in the same time very early during embryogenesis, and they will seed the skin of those fetus. And you find that during controlled pregnancy, fetal skin mast cells are not degranulated anymore. Well, if you analyze the pups, the embryos during a stressed pregnancy, we'll find the exact same phenotype that we found at birth and in the adult mast cell, is that mast cells are already degranulated at steady state. So we wanted to see whether they were also transcriptomically dysregulated. So we sorted those mast cells that you can do. So they express very dim level of the high affinity Ig receptor within the fetus. And even sometimes, actually, we found that they do not express the Fc epsilon receptor 1. So we can sort them easily using the IL-33 receptor and CD117. And when you do that and you do a bulk RNA-seq analysis, you will find that control mice and prenatal stress mice will segregate actually far apart one from each other, with more than 1,000 genes that are differentially expressed on those mast cells coming from controlled pregnancy or from stress pregnancy. And when you look at those genes again, how they are related to, we found that these are genes associated with granule formation, remodeling, maturation of mast cells, but also activation of mast cells. And also, interestingly, we also find some genes that are associated with mechanical stimulation and modulation of sensory perception. So we still don't know the direct link because it's a really complex phenomenon, but between mast cell and sensory neuron, it can be that these are two differentially dysregulated pathways that will not interact, but that they might be also interacting together. So we next wonder actually what could be dysregulating those mast cells during the embryogenesis, during the pregnancy. So for that, we took advantage of public data set published in 2024 of skin cells, actually, in which you find quite good amount of skin fetal cells. You find very good amount of macrophages and mast cells, so you can differentiate the two of them using CPA3 and CSF1R, for example. And we decided to analyze the two of them just in parallel, just to have a readout and a control of cells that would develop in the same time during pregnancy. And we stand for two, we analyze the expression level of two important receptors, the adrenergic receptor, beta receptor 2 for the SAM axis, and the glucocorticoid receptor for the cortisol axis and the HPA axis, which are two very important stress response systems. And when you look at that, you will find that only macrophages and not mast cell express the adrenergic beta receptor 2 within the embryo. So, and during the time that we actually stressed the mothers. And mast cell might eventually acquire it progressively, but after birth. However, when you look at the glucocorticoid receptor, it's the opposite. You will find a mast cell within the embryo. They express very high level of NH3C1, while macrophages actually express low level as compared to that. And again, the level will stay very stable all even after birth in mast cell, while macrophages will acquire it progressively after birth. So we thought, okay, maybe dysregulation through the glucocorticoid receptor during the embryogenesis could lead eventually to mast cell, to reprogramming of those fetal mast cells. So what we did is that we first actually checked that we could find the increase in corticosterone release, which is produced after activation of the stress response system, the HPA axis. And we indeed found that in the amniotic fluid, where those embryos are actually bathing all the time, and we found very increased level of corticosterone during prenatal stress. And if you look at the transcriptome program of those mast cells, you will find that they do have many genes that are known to be associated in response to activation of NR3C1 and of the glucocorticoid response system, meaning that those mast cells within the embryo, they certainly receive direct signal actually through NR3C1. So how we could demonstrate that as well. So we then took a little bit step aside and work ex vivo on fetal skin mast cells. So we sorted fetal skin mast cell and we incubated, we performed the same degranulation assay that I showed you at the beginning with the X2 inhibitor in collaboration with Esciand and Insight, and where we analyze actually degranulation in vitro. And when you do that, you take the fetal mast cell. You can see that when you inject, for example, substance P, as a control, they do degranulate in response to substance P stimulation. So it's possible to degranulate those fetal mast cells ex vivo. Then what was very interesting is that if you put corticosterone alone, it does trigger mast cell degranulation in those fetal mast cells. And if you take the amniotic fluid from control pregnancy, they do not degranulate mast cell. But if you take the amniotic fluid from prenatal stress, it does activate mast cell pretty well. And so we wonder, whether this corticosterone and HPA axis activation within the mother would be a key regulator of this phenotype that we observe. And to understand this, we cannot obviously use, corticosterone deficient animal because they will not be viable then at birth. However, we found a protocol to have a transient inhibition of corticosterone synthesis thanks to repeated injection of metirapone that inhibits the enzyme responsible for corticosterone synthesis in the mother. And when we do that and you measure corticosterone level in the blood of the mother, you can find again, like in the amniotic fluid, that you have this increase in corticosterone. And when you use metirapone, you will find that there is actually no more increase here. These are control mothers treated with metirapone. And so you have a small increase in corticosterone, and we think it's due to the stress of the injection actually in the mother. But when you do that in prenatal stress with metirapone, you can see that it's not dissociable from the control mother. You kind of restore almost physiological level close to the control pregnancy. When you do that, you do not interfere, obviously, with healthy pregnancy. The mother are gaining weight normally. The litter size is the same. And then the weight of the pups, even at birth and after that, is completely normal as well. But we were really fascinated to see that when you treat the mother and you normalize or reduce at least those corticosterone level, you actually fully protect the pups at birth from the development of pediatric eczema. There is no more increase in epidermal thickness, and you also, interestingly, restore a normal mechanical sensation in the in-pouch using the von Frey filament assay. And if you look at the mast cell, which are obviously key driver of the pathology, we found that again, those mast cells are completely back to normal. They are not degranulated anymore. And this is quantified here in prenatal stress when it's normalized with metirapone. And so with that, I'm arriving to the end of the presentation, and I would like to propose maybe a revisited version of the etiology of very early pediatric eczema with remission that would be more centered around in utero dysregulation of neuroimmune circuit. Obviously, this will not explain all the pathology of pediatric eczema, but we think it can explain at least in part some of the features that we found in those infants. We think that chronic psychological stress during pregnancy via the activation of the maternal HPA axis will lead to corticosterone synthesis from the mother. I didn't show you there, but actually it's not the pup that secrete the corticosterone, which are increased level of corticosterone, but it's really the fact that they are bathing in the mother corticosterone environment. This will activate and engage the NR3C1 or the glucocorticoid receptor on mast cell that will trigger degranulation, impairment and reprogramming of those fetal mast cell that will then persist after birth for many months. There will be also dysregulated sensory neuron and mechanical perception. We think it could be bidirectional communication, certainly, that will be dysregulated, and communication between sensory neuron and mast cell, but still, this is difficult to investigate. And this will lead to mechanical hyperreactivity and what we found feature of eczema-like manifestation. And after, when you have the replacement of those mast cells at 6 months by additional mast cells that are not imprinted anymore by this in utero environment, then you can go in remission. So for that, I would like to emphasize that obviously it's really good to chill during pregnancy, and but not chill too much, obviously. And a healthy pregnancy can lead obviously to healthy fetal programming and healthy tissue then after birth. And with that, I would like to thank all the people that participated to the study that I show you there. These involved many people, such as Lilian Basso, Nadine Serran, Marie Taubert. On the Genoskin side, Lynn Pages, Manon, and Lynn Mathias. And obviously all the people, all the collaboration and the precious collaborator of these projects, and all the sponsors that give us some money, some time. And with that, I would like to thank you for your attention. And I hope you enjoyed the talk, and I'm happy to answer all the questions then on Twitter. So thank you very much and have a good day.
Well, thank you, Nico, for the really fascinating and spectacular talk, and for instance, in particular, for showing us unpublished data at the end, which I think everybody enjoyed. Now it's time normally for questions, but as everybody knows, we make the sharp cut here. So, and I have to refer to our Twitter page.
We don't hear you anymore. I think you're on mute, Dietmar.
I've been talking all the time and I was muted, but actually I hit the button,
but I
did not hit it correctly. So I have to do it again. Sorry. Thanks a lot for this really, really fascinating talk. And I think it was also very, very good. And everybody laughed at that we saw unpublished data at the end. Now, I'm sure that there's lots of questions, but as usual, we refer everybody to ask those questions on the page formerly known as Twitter. And with this, we make our usual sharp cut, and thank you very much again for sharing the data.
Thank you, Dietmar. Thank you, everyone, for listening.
And I should also announce that Andreas Wack is our next speaker from the Crick Institute, and so looking forward to seeing you all there again. And other than that, I wish everybody a very nice remaining day or a beautiful evening, and thanks for joining, and seeing you soon again. Goodbye.
Thank you. Bye-bye.
Ciao.